Today's speaker is Professor Andrzej Kierzek. Dr. Kierzek earned his undergraduate degree in molecular biology from the University of Warsaw. He then went on to earn a doctorate in biophysics from the Polish Academy of Sciences. He's currently a professor of systems biology at the University of Surrey in the United Kingdom and is the head of systems modeling at Certara. Andrzej has more than 20 years of experience in computational biology, and his research focus is on immune oncology and immunogenicity. Andrzej, welcome to today's event. I'll now turn it over to him to begin the presentation.
Okay, thank you very much for coming to my talk. As Suzanne said, I recently made two important transitions in my life. First of all, I moved from academia to industry. I used to be at University of Surrey. Now I'm in Certara QSP, which is part of SIMSIP. But the other transition, which some of my neighbors think is equally radical, is that I moved from southeast of England to the north. So University of Surrey is located just outside of London Orbital motorway, and now I work in Sheffield, which already counts as north of England. So this is summary of what I'm going to tell you today. So first of all, I will remind you that DNA sequencing is truly disruptive technology, and that ability to sequence individual person's genome is likely to change medicine substantially. And then I will argue that this actually motivates mechanistic modeling of how physiology emerges from the interaction between genome and environment. Then I will show physiologically-based pharmacokinetic modeling as a motivating example of large-scale mechanistic modeling approach, which already simulates function of about 20 genes. And then I will dedicate remaining part of this talk to saying how this mechanistic modeling approach can be further expanded into intracellular space. And in particular, there is one special case of molecular interaction network, and this is cellular metabolism, where we can already do it to large extent for 2,000 genes. So I will introduce genome scale metabolic networks. I will tell you how we can apply these networks for mechanistic interpretation of omics data, and then I will present case study of how these models can be integrated with PBPK and also dynamic models of gene regulation. So genotype-phenotype relationship. It's fair to say that nowadays we can determine any DNA sequence of interest. In particular, we can determine full genome of individual, and we can also do it for many individuals. We can fully genotype a population of patients. And currently, these data are analyzed directly by statistics, so by statistical inference. So we look for associations of nucleotide polymorphism with phenotypic traits. So this is example of work in this field, and then I had the pleasure to contribute a small part of the analysis. So in this meta-analysis, we managed to collect full genotypes of over 80,000 individuals, and these individuals were in two groups. We had people with prostate cancer and healthy people, and then for each position in the genome, we compiled nucleotide frequencies, and we tested null hypothesis that in this position of the genome there is no difference between nucleotide frequencies into groups. And on this so-called Manhattan plot, you see negative logarithm of this p-value, and this is plotted along whole genome, and whatever stands above this significant skyline indicates genetic loci with association to prostate cancer. And in this paper, we identified 23 new suspect loci which are associated with prostate cancer. And this is, of course, very valuable work, and I'm very happy that I was involved with this to some extent. But we do not know, after doing all of this, what molecular and physiological mechanisms associate these loci with cancer. So we don't know why these loci are associated with cancer. And of course, without the knowledge of underlying mechanisms, it is very difficult to use this information to design new therapies and biomarkers. Also, usually we are interested in much more complex effects than this simple case control design that we were studying here. So, for example, we would like to know effects of combinations of drugs, combinations of toxic substances, diet, lifestyle, and then the question is, can we study this more complex situation by this direct statistical inference, given that even for simple designs we required very large sample sizes to see associations. So of course the alternative to statistical inference is mechanistic modeling, and this approach is of course much more difficult to implement, to conduct, but usually much more rewarding. So I spent much more rewarding in terms of both understanding and predictive power. So I spent a significant part of my academic scientific career on building mechanistic models of genotype-phenotype relationship. So what I would like to see happening is to take the substantial knowledge that we have about molecular cell biology and represent this knowledge in executable form of a computational model. So there is a library, and some people in my field try to rebrand library as a biblion, but it's a library containing peer-reviewed research papers describing individual interactions between molecule A and molecule B. I would like to collect these interactions, represent them in the form of computational model, and then for given genotype and given content of cellular microenvironment, I would like to run simulation and observe dynamics of molecular changes within the cell. And of course, the cell knows how to express its genome in the context of given environment. So if we could reverse engineer machinery that does it, we would gain unprecedented predicting power in seeing and understanding what happens when particular genotype interacts with particular environment. So this, of course, would require mechanistic modeling at the unprecedented scale, and the question is, can we build very large-scale literature-based mechanistic models that would be successful, that would have sufficient predictive power for industry applications? And I think that PBPK, that many people in the audience would know very well, provides a motivating example in this area. So very, very briefly, PBPK describes how the drug is absorbed, distributed metabolites, and eliminated in physiological compartments. The variable of the model are concentrations of the drug in different compartments, and the model has mathematical form of the system of ordinary differential equations, where we simulate time evolution of these variables, and we use blood flows, physiological compartment volumes, and partition coefficients as parameters. Now, these parameters are not directly estimated from the data set that we have for particular study. These parameters come from literature or from in vitro assays. And when we have this model, we can simulate dynamics of the system, in this case, concentration changes in physiological compartment of interest. Now, the models, the PBPK models which are used in contemporary applications are by far more complex than this basic model that I was showing you on the slide. So individual physiological compartments are further expanded. They are divided into subcompartments, additional flows, additional partition coefficients are added, and more mechanistic detail is available. And in the full version of our PBPK simulator, of SimSip simulator, we have models for liver, gut, kidney, brain, and lung, as you can see on this slide. Now, the model of this complexities, of course, has to be provided with tools, and we provide our SimSip simulator software where you can use this model and the knowledge about variability of parameters in population to simulate virtual trials. Now, one of the reasons why I'm showing you PBPK as motivating example is that this is large-scale mechanistic modeling. This is representation of literature and in vitro assays, which is successful in industry. So here you see example of a drug label where PBPK simulation has been used as a sole evidence for the statement that two drugs interact, and for evaluation of the magnitude of this interaction, and of course, because we could use computer simulation here, some clinical trial did not have to be conducted, and this is by no means isolated case, and in fact, PBPK has been used for almost 100 label claims in not only drug-drug interactions, but also absorption, ethnic bridging and formulation changes. So this is example that very large-scale computer model representing literature and in vitro assays can achieve the highest possible level of quality, which is to be suitable for regulatory submission. Now, in fact, PBPK is already simulating genotype-phenotype relationship. It does it for 20 genes, so the model includes about 20 genes encoding drug metabolism enzymes and drug transporters, and in fact you can input genetic polymorphism in your population of interest into simulator and use it in virtual trial simulation. However, there is obviously a huge gap between being able to simulate relationship of 20 genes with pharmacokinetics and being able to conduct genotype-phenotype simulation informed by entire genome, where we expect to see about 30,000 genes. So the question is, can we extend mechanistic modeling approach to account for all genes and also to interpret omics data by simulation rather than statistical association? And this comes down to extending further mechanistic detail of PBPK compartment, which is called intracellular space. So in PBPK model, intracellular space is treated just as a volume. Now we would like to expand it further and see in these intracellular modules individual species interacting through some reactions. Now, in the ideal world, I would like to build this model in quantitative framework, so I would like to have variables which are molecular species, which are described by molecule numbers or concentrations in the cell, and then this species would interact through certain reactions, and then I would use ordinary differential equations or stochastic chemical kinetics to simulate dynamics of molecular amount changes in the cell. The problem with this approach is that molecular biology knowledge is very qualitative. We know a lot about existence of interactions in the cell, but we don't know so much about quantities of molecules in the cell, and we generally don't have sufficient information to parameterize large-scale quantitative models. However, there is one particular case where large-scale mechanistic models already exist, and this is intracellular metabolism. So here, I will not be talking about the drug metabolism. Here I understand metabolism as a set of coupled chemical reactions that the cell uses to convert nutrients available in cell microenvironments into building blocks, into energy, and into reducing power. And the models which are available, for example, the RECON2 model compiling entire human biochemistry, these models can account for about 2,000 to 3,000 genes, and here you see the entire scale of the model like that, as well that on this inset you can see the level of detail. These are really all enzyme catalyzed conversions of small molecules in the cell. And with this model, the following calculation becomes possible. We can perturb some of the reactions in the model to represent, for example, toxic compound, drug action, but also to represent genetic polymorphism, leading to different, let's say, Vmaxes of metabolic enzymes. And then for the given perturbation, we can calculate maximal possible flux through selected reaction in the system. And this reaction represents metabolic capability of interest. For example, what is maximal availability of glutathione, what is maximal production of bile acids, what is maximal production of kynurenine or prostaglandin, etc. Now, being able to do it does not come for free. There are two important assumptions that are being made for this analysis of unprecedented scale to be possible. First of all, variables of the model are reaction fluxes rather than substance concentrations. The second assumption is that we study this system at steady state. So the model is still represented as the system of ordinary differential equations, but now all concentration changes are zeros and variables are reaction rates directly. And this leads to the linear model that can be then additionally constrained by available experimental knowledge on these fluxes. And while we cannot solve this model to determine all fluxes for which we do not have experimental information. What we can do is to calculate maximal value for any linear combination of fluxes. So we can create objective function of interest, such as, for example, maximal biomass production, which would represent the growth rate of tumor cells, or bile acid production, or kynurenine production, or any other metabolite capability of interest, and we can get maximal value. Now, if this possible maximum value is 0, then we gain important information that in this particular a model subject to certain perturbation, this metabolic function is impossible. Otherwise, we have estimation of maximal possible flux, and this method is called flux balance analysis, and is certainly referred to as a constraint-based approach because we calculate maximal magnitude of metabolic capability given number of constraints. So this is my example of actual simulation of genotype-phenotype relationship that we performed with colleagues at Surrey now 10 years ago. So we built the model representing genome scale metabolic network of Mycobacterium tuberculosis, which is bacterial pathogen. And then with this model, we did the following virtual screen. We took every metabolic gene in the genome, inactivated this gene, then we inactivated all reactions associated with this gene, and we calculated maximal value of biomass production. And whenever this value was 0, we concluded that the gene is essential. And this was compared with experimental data coming from high throughput transposon mutagenesis, and we could actually evaluate predictive power of this model in actually simulating genotype-phenotype relationship. I mean, finding essential genes, simulating which genes, if inhibited, would prevent biomass production. And here you see ROC curve, and also you see some parameters, sensitivity, specificity, and these predictions are, this predictive power is certainly statistically significant, and it is certainly very good starting point for use of this methodology. Now, obviously, especially in multicellular organism, there is nothing like genome scale metabolic network of the organism, and the reason for that is that in every individual tissue, liver, muscle, heart, in every individual tissue, metabolic network will be slightly different, and this is because different set of metabolic enzymes will be expressed. So to build tissue-specific models, the ideal approach is actually to try to constrain this general model by transcriptome data obtained for particular tissue, or by proteome data, in general biomics data. And this is done most frequently by approach proposed by Eitan Rupin's group in 2008, where we maximize, instead of maximizing metabolic capability, we maximize congruency of metabolic flux distribution and transcriptome data. So for example, so we look for metabolic flux distribution such that reactions associated with downregulated genes are switched off, and reactions associated with highly expressed genes carry large flux. And here you see the example that to achieve agreement of gene E5 and E6 with metabolic model, we switch off entire pathway, and we have to switch off entire pathway because we require that all metabolites in the network are balanced, that all stoichiometric constraints of the network are balanced. And then we have hypotheses that for gene E7, we either have an error in experimental data or there is some post-transcriptional regulation. And likewise, we see that entire pathway originating from metabolite M1 has to be regulated, and we have hypotheses that reaction for which we do not have any gene expression data, that this reaction actually has to carry substantial flux. We applied this approach, my PhD student Vytautas Leonsikas, who is now working for me in Certara on our IMI2 grant. He generated transcriptome data specific metabolic networks like that for 2,000 breast cancer clinical samples available in MetaBrick dataset. So without going into details, we used Recon2 model of Genome Scale Metabolic Network, the general model, and then for 2,000 individual tumors, we had transcriptome data. And after applying the approach that I was showing you, we had effective metabolic flux distributions for 2,000 individual tumors. Now, these metabolic landscapes could be then clustered, and since for each individual tumor there were clinical data, we could calculate Kaplan-Meier curves for clusters and identify poor prognosis cluster. Since clustering was done by metabolic networks rather than transcriptome data directly, we could look for metabolic features distinguishing this cluster from others. And in this way, we identified that serotonin pathway is going to be active in this poor prognosis cluster. And then we examined literature whether serotonin pathway was actually somehow associated with breast cancer in studies conducted before. And in fact, we see a typical literature in cancer research that some papers actually list serotonin pathway as a potential drug target. There are papers that argue that nothing happens, but we see that this is because this pathway is important in a particular cluster of poor prognosis patients. So this is the example how large-scale mechanistic model combined with omics data can highlight a molecular pathway of interest, and I think that genome scale metabolic networks are ideal for this. We then moved to working on multiscale simulations where we were combining genome scale metabolic networks with dynamic models. And we published free academic software for doing this, and this is work at the University of Surrey. This is not Certara software, this is academic software. And then we moved into combining genome scale metabolic networks with PBPK, and now I will show you a case study that we submitted to CPT as a tutorial. And this is to show you how integration of genome scale metabolic networks with PBPK can work. So we built here the following model, the following case study. We have basic PBPK model, and in this basic PBPK model, we modeled a compound, a drug, which is metabolized in liver and produces toxic metabolite. Now, this toxic metabolite is then removed by conjugation with glutathione. And you can immediately see that the case study we are talking about is paracetamol, but we did not input any specific compound information for paracetamol. This is just a hypothetical model that shows similar behavior. And then in the liver, the compound is metabolized by one of the CYP enzymes, and we have a quite sophisticated dynamic model of regulation of this gene as a function of occupancy of glucocorticoid receptor, which then depends on cortisol concentration, but also as a function of PXR, which has a number of ligands which are used as drugs. And then this in turn is combined with Genome Scale Metabolic Network, and we use Genome Scale Metabolic Network to basically calculate maximal glutathione availability. And so I will now describe briefly the components of this model in more detail. So let's start with Genome Scale Metabolic Network. So we used HepatoNet1 model, which is a general model of human metabolism tailored to liver by combination of analyzing omics data, but also doing dedicated literature searches. By the way, in my personal opinion, this is still the most accurate genome scale metabolic network built for human tissue. What we then wanted to do with this model is to actually make it more quantitative than it is at the moment, because this model was used mostly for qualitative predictions, whether certain metabolic functions, for example, detoxification of certain xenobiotic can happen. We really want here to have some quantitative estimation of an upper bound of glutathione availability. And for this, we used a omics data set which was obtained in the context of a different study. So in 2012, this group published in Science the study of NCI60 cancer cell lines, and for each of these cell lines, they performed measurements of metabolites in the medium, and they did it as a short time course. So basically for 219 metabolites in 60 cell lines, they were able to estimate consumption or release flux. So what we decided to do was we introduced additional assumption of the model. So we assumed that under physiological conditions, none of the nutrients is consumed or released by human hepatocyte with higher flux than the flux observed among 60 fast-growing tumor cell lines. And this is because these are cancer cell lines, therefore they would divide faster than any healthy tissue, and these are also cell lines, so they divide actually faster than tumors in patients. So if these cells with high metabolic demand do not consume something faster than certain upper bound or slower than certain lower bound, we can use these values to bound transport reactions. And then we were able to calculate maximal glutathione availability given a full boundary conditions limited by this in vitro data, and the values that we were getting had much more now quantitative interpretation. So the first thing that we did with the model was virtual screen. We inactivated each reaction one by one, and then we collected all reactions where this inactivation changed maximal glutathione availability. And then here you have list of reactions. I didn't put here the number of reactions. I think there are about 50 of them. So this is an example how we can identify reactions where pharmacological inhibition, toxic compound, or in fact genetic polymorphism can make a difference on metabolic function of interest, in this case glutathione availability. So for PBPK model, we used very basic model from PSP tutorial published by Hannah Jones and Karen Yeo, and I don't think I'm going to talk a lot here about PBPK. I already introduced this modeling, and I assume that a lot of people in the audience are familiar with this. Now, for gene regulatory network, we took a typical systems biology model. So this is the model published by a group of my colleague Nick Plant and Hans Westerhoff, and they built ODE model of CYP3A4 regulation by GR and PXR receptors, and here we can study the changes of gene expression as a function of cortisol and PXR ligand. And then we combined these three models together, and we did it in following way. So GSH availability was calculated as an objective function of flux balance analysis. So basically we used maximal flux of GSH as an input, as a parameter to GSH conjugation reaction. Now, the toxic compound itself, we assumed that it inhibits one of the reactions in genome scale metabolic network, where we had some evidence that it was again based on paracetamol, that paracetamol metabolite can block this reaction. And then for the rate of metabolism of the drug into toxic compound was parameterized by relative level of CYP3A4 protein. And with all these models combined together, we could now run dynamic simulation, and here you see different cases. So for example, inactivation of two reactions which are listed on figure A would increase maximal concentration of toxic metabolite. There is one reaction which you see on plot B, where inactivation of this reaction would lead to very high, probably unrealistic increase of toxic metabolite concentration, but this reaction should be of special interest to anybody working in this drug, because of course polymorphism in this reaction could lead to population of people that actually shouldn't be given the drug because the effect of toxic metabolite would be particularly severe. On plot C and D, you see the PK as a function of endogenous metabolites. So we give bolus of cortisol, which represents stress event, and again you can see, you can study PK of this metabolite as a function of cortisol, and this is now informed by a very sophisticated model of gene regulation. So in summary, I would like to leave you with the following messages. That I think that personal genome sequencing very strongly motivates mechanistic modeling of molecular cell biology. To fully use information about genetic code of the patients, we should use full knowledge about molecular cell biology, and this knowledge is so extensive and interactions are so nonlinear that to fully use available knowledge, we need mechanistic models of molecular cell biology. Now, the genome scale metabolic networks are available for very special case of metabolism, and we can already model thousands of metabolic genes. This is an ideal tool to interpret omics data, and then the PBPK models, which have been used in the industry for very long time, already model a function of about 20 genes, and integration with genome scale metabolic networks can expand the scope of pharmacogenetic modeling. And then further integration with gene regulation models with systems biology will enable to study interactions with endogenous metabolites. So in the final conclusion of this talk, I think that mechanistic modeling of molecular networks covering function of thousands of genes will soon become an important tool in model-based drug development. I would like to finish with very quick acknowledgements to a lot of people who worked with me on this. So you see that that our academic software resulted from international collaborations of many colleagues, and also I worked with people both in Surrey and ICR Saturn on cancer and liver, and then I had very long-standing collaboration on tuberculosis, and I'm very happy that I was funded by funding bodies which are shown here. So with this, I will finish. Thank you very much, and I'm happy to take questions.
All right, thank you so much, Andrzej. We'd like to invite our audience to submit their questions to Andrzej in the Q&A panel. Looks like we've got our first question from the audience. Your talk focuses on pharmacokinetics. Are genome scale networks applicable to simulating drug action?
Yeah, so obviously I focus this talk on pharmacokinetics because this is currently our work, and we have this case study, a kind of virtual screen that I was showing you for essential reactions for particular metabolic function. This could be applied on discovery stage of drug development to find targets, but also when we know target, and this is the enzyme, this type of models can be used to estimate the change of maximal availability of the substance upon inhibition. So by all means, these models can be used to simulate drug action.
Is flux balance analysis applicable to cell signaling, or is it limited exclusively to metabolic networks?
Okay, thank you very much for this question. I mean, this is actually something that I did not have time to talk about. So in the case of, let's call it forward signal propagation through signaling network, we can directly use flux balance analysis to study. We can basically evaluate whether we will have certain output signal distribution given certain input signal distribution. The problem starts when we want to introduce feedbacks, when some molecules in the network are part of negative feedback loops, because this is not very well modeled in stoichiometric framework. Now, in our software, we did design dedicated linear programming approach to model negative feedback loop. This is based on earlier work on Eitan Rupin again, but our software is the first one that provides interface and language of this. So if you read our paper about multiformalism interaction network simulator, you will find case study where we model signaling network of a macrophage coupled to genome scale molecular metabolic network of a macrophage, and the output of interest is maximal nitric oxide production as a function of signaling network inhibitors.
Someone would like to know, do we have to consider the SNP and epigenetic modification of the metabolic genes?
So Genome Scale Metabolic Network itself does not provide methodology of estimating the effect of genetic polymorphism on enzyme activity. So this has to come from somewhere else, either from, I don't know, in vitro data or from molecular modeling. Basically, it also depends on experimental information that we have. If we know only sequence, DNA sequence, that the only thing that we can take into account is, of course, nucleotide polymorphism, and then we have to find a method to estimate the effect of this nucleotide polymorphism on Vmax of metabolic enzyme. However, if we have information about methylation pattern, about epigenetic changes in particular genetic loci, then likewise we can take it into account.
Someone says, bioinformatic tools for transcriptome data analysis highlight upregulated metabolic pathways. What is an advantage of using genome scale metabolic networks for transcriptome data analysis?
So in fact, we tested this in our publication about metabric data sets. So we took transcriptome data from the original paper and we applied David annotation tool, which we consider to be state-of-the-art in identifying overrepresented descriptors in transcriptome data, and we could not recover pathways that we identified with genome scale metabolic network approach. Now the reason for that is that frequently only one gene needs to be differentially expressed to upregulate entire pathway. And this is simply not enough to pass hypergeometric statistical test threshold and flag entire pathway as upregulated. Whereas with this approach, because we require that no substance accumulates or appears from nowhere, if you change capacity of one reaction in the pathway, entire pathway has to change, and then this is visible in our analysis. So in our paper we show examples of why this global approach is better.
Someone would like to know which therapeutic areas would you guess would be the early adopters of this type of modeling? Oncology seems obvious, but it's also very broad.
Yes, so I think that in fact oncology is the obvious area for two reasons. One of the reasons is obviously that traditionally metabolism is a drug target because cancer cells, they have much larger metabolic demand than healthy cells, and there are actually quite a lot of academic papers at the moment where authors analyze, use genome scale metabolic networks to find metabolic vulnerabilities of cancer cells, and I think that the progress is being made in this field. But I think that another field of oncology where this can be applied is immuno-oncology, because quite a lot of metabolites such as kynurenine or prostaglandin are in fact immunosuppressants. So then thinking about how genetic changes or how changes within tumor cell affect production of these metabolites, or what would be optimal drug targets or combinations to inhibit production of these metabolites. I think that this is our application area. What we are doing here as a part of IMI grant, we apply genome scale metabolic networks in context of toxicology, in particular drug-induced liver injury, so this is another possible area. Then perhaps a little bit more, I mean certainly much more sophisticated application is in the field of health advice for obesity, for diabetes, and in particular non-alcoholic liver disease. So we could look for genetic polymorphisms that make people vulnerable for metabolic syndrome, for example, for this metabolic diseases. I think there is application there.
This next question fits in well with your comment on immune oncology. Metabolic genes are interact highly with immune genes. Do we need to consider this?
Well, so in the ideal world, I would really like to have a model of whole cell where we would consider entire molecular biology of the cell. And of course, our world is non-ideal, and we are limited in what we can do. Nevertheless, as I said in my software paper, I provided demonstration model where we built a signaling network of mouse raw macrophage, which is the model in immunology, and coupled this with genome scale metabolic network, and then you can see actually very indirect interactions between signaling networks and metabolic genes, because, for example, a signaling network inhibitor, it was MEK inhibitor that we used both in the model and experimentally, blocks signaling pathway that activates enzymes involved in nitric oxide production. So we could predict, we could simulate the changes in nitric oxide production as a function of signaling network inhibitors. And of course, our model already has potential of studying interaction between signaling network and metabolism. So by all means, I would like to make the model as close to whole cell model as it is possible given state of the art.
Looks like we have one final question from our audience. Someone says, kinetic models are available for many metabolic pathways, including important drug targets such as COX-2. Why use genome scale metabolic maps instead of these models?
So the advantage of kinetic models over flux balance analysis is that they are dynamic. However, the disadvantage is that they have very arbitrary boundary conditions. So, for example, if you want to have, let's say, kynurenine pathway, it needs to start at some precursors in the cell, and you have to assume availability of these precursors as your parameters. On the other hand, with flux balance analysis, we can put these cell physiology bounds that I described, and then use stoichiometry of entire metabolism to calculate quantitatively what is maximal kynurenine availability. And both approaches actually carry severe assumptions, and it may well be that actually whole cell approach gives more realistic picture. Another advantage of this global model is, of course, that then you can perform virtual screen and identify all reactions that affect synthesis of kynurenine, and actually there may be some, under particular conditions of nutrient availability, there may be some surprises there. It's not necessarily only IDO that would affect kynurenine availability.